DNA replication is a multi-step process. The enzyme topoisomerase starts at an origin of replication and unwinds the two strands of DNA. The job of the helicase is to break the hydrogen bonds in between the two strands, creating a replication fork. As helicase is breaking the bonds, it forms a replication bubble. SSB, or the single-stranded binding proteins, are used to hold two DNA strands apart, which prevents the DNA from binding back together after the helicase unwinds it.
After helicase breaks the bonds and forms the replication bubble, the enzyme primase is needed to start the replication of DNA. It lays down the primer made of RNA at the origins of replication along the eukaryotic strands of DNA.
As you can see, the two separated DNA strands are anti-parallel, with the 3' to 5' strand on the top and the 5' to 3' end on the bottom. DNA polymerase III builds DNA bases from the RNA primer laid down by the primase from the 5' end to the 3' end on both new strands to make sure it also runs anti-parallel to the two original strands. As the helicase continues to move and break the hydrogen bonds, the leading and lagging strands are formed. On the top is the leading strand because the DNA polymerase III will follow the helicase to make DNA without any interruptions. On the bottom is the lagging strand because as the helicase continues to move, new primer has to be laid down in order for new DNA fragments to be built.
DNA polymerase I is an enzyme that bonds DNA by removing the RNA in the primers used during replication and replacing it with DNA. Additionally, this enzyme will check to make sure the nitrogenous bases are correct and will correct any incorrect bases in the new DNA strand to prevent any mutations that could correct or cause incorrect processes like protein transcription to occur.
As the lagging strand continues, small segments called Okazaki fragments are formed. The DNA ligase takes those Okazaki fragments and glues them together by creating phosphodiester bonds between the fragments to create a fully continuous strand. DNA replication occurs semi-conservatively, where the original double strand separates and each original strand in red has a new strand in green built onto it. This continues throughout replication.